Welcome to the Molecular Therapy Podcast. I'm Dr. Rory Bricker-Anthony, Scientific Editor of the Molecular Therapy family of journals. In honor of Molecular Therapy's 25th anniversary, this episode is the second in a series hosted by Dr. Joseph Glorioso, Editor-in-Chief of Molecular Therapy, that will cover comprehensive reviews of critical developments in the field of gene and cell therapy over the past 25 years. In this episode, Dr. Glorioso will discuss the reviews AAV Vector Development Back to the Future with authors Sheila, Mikhail, M34, and The Delivered Promises of Gene Therapy, Past, Present, and Future of Liver-Directed Gene Therapy with author Dr. Mark K., Stanford University. If you enjoy this deep dive into research that is critical to the field, check out two more ASGCT events happening this fall: Advancing Cell and Gene Therapies for Cancer and Breakthroughs in Targeted In Vivo Gene Editing. During these events, in October and November, respectively, you'll be able to hear directly from researchers about their own work related to these topics, or you can submit your own abstract for possible presentation. Visit asgct.org/advancing or asgct.org/breakthroughs for more information. We hope to see you there. And now, Drs. Glorioso, Mikhail, and K.
Okay, hi everybody. I'm so glad to see you, Mark and Sheila. We're lucky today. We have two of the world's experts in the development of adeno-associated virus vectors and gene therapy. So it's really a pleasure and an honor to be here with you. They have written two more amazing reviews for the 25th anniversary issue of ASGCT Molecular Therapy, and these reviews are really worth reading. They're very comprehensive and they cover the fields beautifully, both the history, the development, and the future. So I'm going to have you guys introduce yourselves, and then we'll get rolling. By the way, my name is Joe Glorioso, and I'm the new incoming editor-in-chief of Molecular Therapy. Mark, you want to introduce yourself?
Yeah, so my name is Mark K. I'm the Dennis Ferry Family Professor in Pediatrics in Department of Genetics, head of the Division of Human Gene Therapy and Pediatrics at Stanford, and I've been working in the field of gene therapy since about 1990. I've worked on many vector systems over the year with the early focus on liver-directed gene therapy, and the review that we were asked to write was on the history and future of liver-directed gene therapy for the treatment of disease.
Yeah, thank you. Sheila?
Hi, I'm Sheila Mikhail. I'm a serial biotech entrepreneur. I am filling in today for Jude Samalski. I am often his sidekick, and I think over 25 years I have learned a lot about AAV vicariously through our joint experiences in starting several companies together. I look forward to contributing to this conversation today.
All right, thank you. So one thing that we could do to lead out is to focus on one of you, if that's okay. And if Sheila, you want to start, you're the lady here. So, and really give us some background between, about your work with AAV and the work that you and Jude have done together, and then talk about maybe some of the highlights that you think are important to mention.
Well, I met Jude serendipitously back in 2001. I was a guest lecturer at a course at UNC called Launching the Company. Actually, I was not supposed to even teach that day, but a lecturer had gotten ill, and I was asked 2 hours before the course started if I would step in and give a 3 hour presentation on how to spin out companies and negotiate with the tech transfer office. But anyway, it was a good friend of mine who taught the course, so I went down to UNC and gave the lecture. And Jude Samolsky happened to be in the audience. He was there because one of his PhD students, Stephen Snowdy, wanted to get a business degree in addition to his PhD. And so Jude had to audit the course to make sure it had enough scholarly merit to warrant the credit. After the class, Jude came up to me and he asked me if I would help him spin out the company. At the time, there was a nuclear winter in AAV. It was shortly after the Jesse Gelsinger death. And even though that had nothing to do with AAV, it was an adenovirus, the shared name unfortunately caused a cloud to go over the AAV field. And it was virtually impossible at that time, I'm sure Mark knows this, to raise any funding. And so Jude, he was definitely a visionary and he was very persuasive. And he basically told me that it would be very difficult to start the company to get funding. But when we were successful, he didn't say if, he said when, we would be able to change the world. And I think that everybody in the AAV field together, collectively, we have changed the world. We have enabled the blind to see, the deaf to hear, and the lame to walk. And we have saved countless lives. So that's how I got started. And at the very beginning, as you know, we were really focused on the original AVs, AV2, AV9, AV8, and some of the re-engineering had begun at that point in time, trying to increase the tropism of the vectors for particular tissues. In particular, Jude was very focused in the beginning on Duchenne's muscular dystrophy and hemophilia. For Duchenne's, he knew that we had to improve the vector tropism for muscle, and he started playing around with AV2 backbones, adding 5 amino acids from AV1 in order to increase the tropism for muscle. And he was a funny guy. He would do all sorts of things like cross-dressing. His papers would be called cross-dressing the virion, which I thought was interesting because it was an interesting way of describing what he was doing. But that's how we started in the very beginning, focusing really on hemophilia, Duchenne's muscular dystrophy, and trying to figure out how to get funding for this emerging science.
Yeah, thanks. So I remember the early day. Jude was an assistant professor in Pittsburgh when I had just arrived there as a department chair. And I can remember we were part of a program grant together, and he came over and he said, "There's no stopping this thing," because he'd just done some brain experiments and got beautiful expressions. So it was a very exciting time. Then he went to Chapel Hill, right? So, but I think the discovery was so exciting because, in the old days, we called AV the Dependo virus, right? Because it was discovered as a sort of a contaminant with adenovirus. And it required genes from adenovirus in order to replicate and produce AV. But I can remember that Ken Burns and Nick Musishka. I know Nick made mutations all over the genome trying to find ways to focus the infection and so on. But it has had an amazing impact on the field of gene therapy, and so many opportunities now have arisen. Maybe you can describe some of what you think are the breakthroughs in engineering the virus and making it more useful for other applications.
I think the first thing that I would like to draw attention to is the hybrid, which essentially took the ITRs from AV2 and allowed other serotypes to be inserted. I think that was a key breakthrough for the field. Second key breakthrough, I believe, was the self-complementary vector, which allowed us to decrease the amount of virus required for therapeutic effect. In fact, for Zolgensma, which is now an approved drug for the treatment of spinal muscular atrophy, it allowed the amount of virus to be decreased by a whole log, and that allowed us to have therapeutic effect while being able to obtain a safety level. And so I think the field continues to work on increasing tropism for particular tissues and also trying to minimize the amount of virus that's required. It's this trade-off. Unfortunately, if the virus isn't, if AAV isn't potent enough, then we have to use more of it, and then we start really budding safety concerns. And so I think that's the one thing that we have done, Jude in particular, over the years for all of our therapeutics, is figure out how do we re-engineer the capsid in a way that allows us to have tropism for our particular tissue, make sure we don't have expression where we don't want, like the liver, try to minimize the amount of expression there, but also try to figure out how to decrease the amount of virus used so we can get below that safety threshold.
Great, thank you. Mark, she mentioned liver. That's your specialty, or one of your specialties.
Yeah. Well, I started, I was an MD PhD student, and my graduate work was on Drosophila genetics. And towards the end of my doctorate work, P element transformation, being able to make transgenic fruit flies were first described. And I actually delayed going back to medical school because there was an experiment I wanted to do with that. And I learned how to do it. And while I was doing these experiments, I kept thinking, wow, this would be so great. This is again in the early 80s. Would be so great to be able to do this in people and treat, genetic diseases. And I thought, well, by the time I finish med school, postgraduate training, this and that, all the interesting diseases would be treated already because the concept of
Yeah.
DNA would be, it was so simple. Well, ultimately, I ended up deciding to do my postdoctoral work in gene therapy. And at the time, we were using retroviruses and adenoviruses. And I actually met Jude. I remember I was a postdoc, and he came and gave a seminar at Baylor College of Medicine. And that's the first time I had heard about AAV. And, when I worked on adeno and retroviruses in the liver, retroviruses, the efficiency of gene transfer was extremely low. It required stimulating the liver to divide or for the cells to divide. And adenovirus turned out to be very efficient at gene transfer, but there was lots of toxicities associated with it. When I moved to University of Washington as a faculty, we continued to work on those two vectors. And I met Richard Snyder when I went to a trip to a company, Somatics, which is no longer around, to talk to people about an adenoviral collaboration. But I started talking to him about AAV. I had gone to Barry Carter, who was at Targeted Genetics at the time, and asked him, because we we're both in Seattle, and he said they really wanted to stick with CF. So Richard and I started a collaboration, which showed, I think, for the first time that, in a mouse, we could get curative levels of factor IX. There was no toxicity. It seemed to be long lasting. And that was kind of the aha moment in my mind for hemophilia. Around that time, we were developing this. Somatics kind of went away, got bought out. The company that bought them really wasn't interested in genetic diseases. So
This was
I
Richard
moved, and I
Mulligan, right?
moved to right, exactly. But Somatics was Richard's company, but it got bought by Cell Genesis, and I moved to Stanford, and I knew Kathy High's group was working on hemophilia as well. And what we ended up doing was working together rather than competing on the hemophilia trial. And like Sheila had mentioned, we decided to go to the RAC and propose. We actually held it back a cycle because Jesse Gelsinger had died. But when we submitted the proposal to RAC and FDA, there was a lot of emotional worry because of adenovirus. And like Sheila said, the names were so similar. In fact, a reporter was talking to me at one point and said, well, you named it so close to adenovirus. That's why. I said, well, I had nothing to do with naming it because I was a few years old when AAV was first discovered. But I try to point out it's like saying, Tylenol and ibuprofen, they both treat headaches, but they do it by very different mechanisms. And I tried to point out that the viruses were really, very different, and the toxicity profiles were different. But there was a lot of pushback from the RAC at the time about using AAV because people were so sensitized because of what happened with Jesse Gelsinger. And, we eventually did do the liver trial. And unlike all the other species, we ended up getting a therapeutic level, but it didn't persist. And it turned out to be the whole issue with the immune response to the capsid once the AAV was in the liver that eliminated the transgene expression. And ultimately that led to the idea of using transient steroid therapies. In fact, St. Jude's moved on from AAV2 to AAV8. We had stopped our trial. The company at the time was Avogen, and it took 3 years to treat 8 patients back then because of all the concerns and things we had to do along the way. And it was longer than the half-life of the company. And St. Jude's and University College London ended up doing AV8, and Kathy and I would go there, and we talked about whether whether AV8 and AV2 would give the same immune response. We actually had invited immunologists, and 5 immunologists gave us 10 answers because we just didn't know what would happen. And I was very opposed to initiating steroids at the outset. I wanted to wait to see if they developed an immune response. And we wrote into the protocol that if the LFTs or the liver enzymes started to go up, the steroids would be initiated. And I think it was useful to learn about this back then.
Absolutely. By the way,
Subsequently over
just
the
for
years,
the audience, the
yeah, go
RAC is
ahead.
the Recombinant DNA Committee at the NIH, which
Yeah,
reviews gene therapy protocols.
Now
right. And
extinct.
the RAC started actually back from, it started with recombinant DNA because back when plasmids were first being made, people were concerned about making genetically modifying bacteria or other organisms that would take over the earth. And
Right.
so the RAC had a early, and then it moved on to gene therapy. And actually, when I was president, I was trying to get rid of the RAC. I thought they'd serve their usefulness. No, I
Right.
think they were great for time. And they would, one of the issues is they always would say they're not a regulatory body, but they were in essence because no IRB at the universities wanted to go against the RAC because if something happened. And sometimes, like when we presented our liver trial the first time, there were some disagreements between the RAC and the FDA, and yeah.
So there's so many liver diseases that are potential targets, single gene mutations and stuff like that. How many diseases in the liver do you think will be amenable to treatment in the future? Probably thousands.
I think potentially thousands, yeah, but it, there's it hasn't gone, I think, as quickly or as the success hasn't been quite as great as one might have imagined because even though many of these are single gene disorders, even within a specific category of an inborn error, many of these diseases have different pathophysiologies. I mean, there's some diseases, like if you take factor IX, it doesn't matter if 1% of the cells make a lot of protein. It gets secreted in the blood. It's all modified, post-translationally modified efficiently enough to become biologically active. But there are other diseases for which the number of cells that are actually being genetically modified is more important probably than the absolute amount of protein. These are enzyme deficiencies that are involved in different metabolic cycles.
Like tyrosinase deficiency? Or?
Not so much tyrosinemia, but things like ornithine transcarbamylase deficiency, the one of the first steps in the urea cycle, is a good example. There's a lot of genetic data that you need a certain, a high proportion of cells to be genetically modified to really cure that disease. And one of the other issues with AAV is that it's primarily an episomal vector, meaning that if the cells divide or the tissue grows, you'll lose expression over time. So many of these disorders really require early treatment in after birth or in early infancy or childhood, at which time you may lose the transgene expression, unless now you can use the AAV to do genome editing in combination with other
Right.
types of moieties like messenger RNAs and to express different nucleases or base editors, things along those lines.
So, do you think the future is going to be treatment of children at a very early age?
I think for many of the inborn heirs, the answer to that is yes. And I think that the use of AAV and other moieties like messenger RNAs and different types of RNA molecules together, perhaps with AAV, will be used to do genome editing early on after birth to treat those diseases.
So,
I think that's going to be the future to really cure those. Yeah.
so I'm curious about the work that's being done to understand what the host range is for these viruses, and I know a lot of changes have been made, mutations in the capsid and mutations, and selections in vivo for certain tissues and all of this. It's been very exciting to see that be developed. I was wondering if this was a receptor driven, which I doubt, specificity, but rather it must be the problem of uncoding and nuclear entry. Do either of you have some thoughts about that? For example, have people taken cells that are susceptible to the virus and done, CRISPR analysis of all these genes and see which ones are really critical for the specificity of the host?
Well, I can comment on that because we've published 2 papers on this recently. Back in the day when Dirk Grimm was a postdoc in the lab, we did interspecies molecular shuffling of AAV vectors and used the library approach to select. And one of the vectors that came out of that was DJ and subsequently Leszek Lazowski as a postdoc. We did in vivo selection in a humanized mouse model. So in these mice, they are chimeric for human and mouse hepatocytes. And a number of capsids came out of that, including LKO3, which has been used in 3 clinical trials. And it appears to work better, let's say, than AAV8 in terms of transducibility. But interestingly, it never worked in rodents. It's 100 to 1,000 times less efficient in rodents. In fact, when Spark did their initial factor VIII trial, that's what they used. And they had to use a surrogate capsid for their murine studies to test their transgene, their factor VIII transgene. And, I had always assumed that it was going to be due to receptor entry into the murine cells. But when we did those studies, and this was published in late 2023, we found that the LKO3 DNA delivered genomes get into the nucleus equally well as, let's say, AV8 or DJ or other capsids. And it even becomes double-stranded, but it doesn't express. And we looked at the histone modifications and found that there's different histone modifications based on the capsid. In fact, if we change the LKO3 capsid by 1 amino acid, we now get expression in rodents. And the histone marks are more permissive, like other capsids. So what we've learned is that the capsid itself is actually participating in setting up the AAV vector epigenome.
Wow, that's very exciting.
But I think it also demonstrates another issue, and that's the translatability across different species. We've all, I think, experienced issues either on dose or just in terms of tropism, where we see something in a rodent model and then it's confirmed when we get to a non-human primate. But when we go into human clinical studies, we have surprises. Either the potency isn't what we expected, and so we have to change the dose, or the tropism, especially in the liver, right? There's multiple different cell types in human livers. The tropism wasn't quite what we had anticipated. And so I think that's something that's very seminal about the work that Jude and his team and the M34 group are doing, which is now basically using human to human translatability, using human decedents to take capsids and screen them to see the tropism and find out, in a human species, what capsids go where. And I think this is a breakthrough because we're continually guessing when we go from the traditional approach of, screening our capsid libraries in rodents and then going into non-human primates and hoping that what we saw in the lower animal species translates in the clinic. We also do just the opposite. We take what we find in our H2H platform, those capsids that appear to have tropism for different human organs, and we translate it backwards. So we go take the capsids that look promising, take them into non-human primates, and then again, if they hold up, take them back into rodent models. I think this is a better way of trying to figure out what's going to work in human clinical trials than extrapolating from a rodent model forward. But again, we'll see what happens. And interestingly, when we did this work, we have now 3 human decedents, which we have screened and characterized the capsids. We also found some capsids that existed de novo. They weren't part of our library. And so hopefully we'll be able to validate that these are naturally derived human AAV capsids. And I hope this, is able to advance the field, and I hope we can share our results with others. Because again, AAV has a lot of promise. And there's a lot of diseases, especially when it comes to the brain and muscle, where AAV has a unique competitive advantage relative to other delivery platforms like LNPs. And so we have to overcome some of the hurdles that we've been experiencing in order to make sure we get the full potential of the AAV, platform. And I believe that based on what we've seen on this H2H platform, that we might have been able to break another barrier to really making sure we can exploit AAV therapies.
So there's been some work using antibodies to target AAV, like the spy catcher system. And I guess based on this discussion, that may not help in the sense that it's really what happens to the virus after the DNA gets into the nucleus that really determines whether it's going to be expressed. Do I have this right or?
Well, I would say that each of these are probably interactive, but they're 2 different variables. I mean, we have to be able to get the AAV into the cells, and then, I think a second parameter is getting it transported to the nucleus and uncoded and expressed. And there's 2 things I want to point out. And this was first, I think, demonstrated by Hiroyuki Nakai at Oregon. But when he used a small library of capsids and he injected them and he barcoded them both in so you could look at DNA and messenger RNA expression. There were capsids that gave discordance. So you can get capsids that get into a particular tissue really well based on DNA, but the expression's low. And I think that indicates possibly that it's some sort of epigenetic phenomena. But I think it points out the complexity and what one really has to look at to get something successful. Because if the DNA gets in at a high rate, but it's not expressed, it's not going to be that useful. And I think another big parameter in the clinical trials is the variability between individuals. So we can only give it once right now because of the immune response. And if you look even at the like factor 9 or the factor 8 trials, for example, where a fairly large number of patients have been treated, there's a huge dose response variance. And BioMarin published recently where they biopsied a handful of the patients like 2 to 4 years after. And there was one patient who never really expressed factor 8. This was using AV5. And when they looked at the DNA amounts between the patients, they were very similar, even 2 to 4 years later. But this one patient didn't express the messenger RNA. So that indicates then, these are all different variables that we need to look at as we develop these new vectors like Sheila had mentioned.
Yeah.
So
The
one thing
bottom
we've been
line is how much expression. Yeah.
So I agree with you. RNA expression is really key. One thing that we have been looking at with regards to the heterogeneity of different patients and the way that they respond to the same AV therapy is the collaborative cross. And the collaborative cross, which is available at UNC, essentially takes all the different mouse lineages and crossbreeds them, gets multiple variations, and it's able to allow us to more precisely characterize who is going to be responsive for a particular AV gene therapy treatment. We need to do more of that because, like you said, Mark, there is so much heterogeneity among different patient profiles that we just now sort of group everybody together and we expect some kind of result. But that's just, I think, very naive thinking. We need to figure out more tools that lets us stratify our patients so we know who is more likely to be responsive to a particular gene therapy approach. And then we hopefully, as a community, will have a lot more successes, in our track record.
No, I totally agree with that. I think we use inbred strains of mice, so we get really close air bars, and then we go into heterogeneous either NHPs or ultimately humans. And if we can find ways to make predictions about how well an individual responds, perhaps based on genetic variances or other variances. I mean, it may not all be genetic. Then we can, like Sheila, like you mentioned, determine what dose a particular individual should get to get a therapeutic level.
So what kind of changes do you see the capsid can introduce into the epigenetics of the genome?
Well, that's something we're studying. I mean, we're trying to understand them, so the thing that we show in the paper is that based on the capsid sequence, you can get a different histone profile. That is, you can see some capsids in certain species will give you a higher percentage of permissive histone marks, ones that are well known to be associated with transcription. In our hands, the repressive lists don't seem to vary as much between, high responder and low responding capsids. But there's got to be molecular events that lead up to this. So these are like end functional biomarkers, if you will. And that's something we're studying and trying to dissect out. And like you mentioned, we've done CRISPR screens, we've done mass spec pull downs, we've done apex labeling where you can unbiasedly label the genome with proteins that associate at different times in the transduction event. And we do have some potential candidates that we're looking at to see, maybe they're druggable or things like that. I mean, in my mind, if you look at the capsids that work really well in mice, and we look at the transcription per genome, and we take ones that work well in non-human primates, and we look at transcription per genome, the transcripts per genome in the best mouse examples are always higher than the best primate examples. And there's very little human data. So the question is either we haven't found the best capsids for humans, or that the molecular events that occur are not identical between, let's say, rodents and primates. And understanding that may give us clues as to how to circumvent those limitations.
That's really amazing. So do you think capsids or parts of capsids actually bind to proteins that regulate epigenetics? Is that what you're thinking?
Yes. And, you know, as the vector uncoats, I think that how the encoding occurs and what the capsids are interacting with. I mean, AAV does uncoat primarily in the nucleus.
Right. And there's also
So,
another complexity. Jude has published on the interaction between the capsids and the ITRs. In some ways, I think, we're using the ITRs derived from AAV2 and sort of forcing that always to be the answer. If we were to change the ATRs to represent the other serotypes, there may be different results that occur too. Clearly, there is interaction between the capsids and the ITRs, and that creates another level of complexity.
Yeah. So I have a sort of a different angle on this. What happens for a lot of these genetic diseases where there's a small population of patients that pharma is not that interested in taking these on because it's not commercially viable, right? How do you think the field is going to deal with this? Because we have to, if we're going to create gene therapy, which is a real breakthrough in treatments, we have to find a way to make it available to more people. And I was wondering what your thoughts are on that.
Maybe I can go first. So when Jude and I were running several companies, which they were bought, two of them were bought by big pharma like Pfizer and Bayer. We tried to tackle this problem by telling the big pharma that they should develop, therapies, AAV therapies for some of these ultra rare indications. And the answer always was, we thought, well, they had more financial resources, so they were the best suited to be able to advance some therapies for these ultra rare indications alongside the bigger population drugs. But we always got pushback because fundamentally for-profit structures are trying to maximize shareholder wealth, and they can't make the business case. They can't get an adequate return on investment for the ultra rare indications. The only way we could see advancing therapies for the ultra rare indications was to set up a foundation, a nonprofit organization. So we set up Columbus Children's Foundation, CCF, which was funded primarily from the shareholders of AskBio through our various transactions. We've helped support Chris Bankowitz's work on amino acid decarboxylase deficiency. Chris has now treated over 50 kids with this devastating disease and has had phenomenal results. Kids who couldn't even turn over before treatment, a year after treatment, are going to school, they're talking, walking, dancing, playing baseball. They're living much fuller lives than before treatment. We also helped form Elpida and help fund Elpida. Elpida is a B corporation. It has gotten substantial funding from CERN and is addressing diseases like SPG 50. These efforts are helpful, but they're pretty small. There's 7,000, 9,000 rare diseases. Many of them are smaller patient populations. It's very difficult to find a way to advance therapies for this ultra rare community. And I think, unfortunately, many of us have advanced therapies for larger indication on the backs of these smaller indications. But when it comes to finding a way to develop treatment so we don't leave, kids behind that just happen to have bad luck of having a disease that isn't shared by many, I don't think we've been very successful. So, we have tried our efforts. I'm always curious and, continuously willing to partner with others who may have some other ideas, because it's very unfortunate that we don't work on more of these ultra rare indications. We have the technology that really could better people's lives and their communities. And it's just, money, right? It's all about money, not having the financial resources to be able to advance therapies for these kids. Many of them are, diseases are suffered by kids.
The
Mark, what are your
research is
thoughts?
expensive.
So can
Oh, go ahead,
Yeah,
Mark.
so I think it's UCSF and maybe UCS Berkeley and some other institutions where I think they're focusing on at least initially hemopoietic derived genetic diseases where there's like an N of one or two, and what they're trying to do is use a genome editing approach. And what they're trying to do is get some sort of blanket approval so that they basically have the nuclei they're going to use to edit the whatever site it is in the genome for a particular disease gene to correct a disease where there may only be an N of one or two. They have to act quickly because the disease could be lethal. And what they're trying to do is get some sort of blanket regulatory approval so that they could just switch out the guide RNAs without
Yeah,
having to go through a whole FDA approval.
yeah,
And the question is, how to go about this. And I think there was a lot of interest in this from Peter Marks and the FDA at some point. Of course, now who knows? But and it's not without risk because we don't know enough about if you change a guide RNA, could you get some off-targeting bad event? But if you limit these to really rare diseases for which they're going to be lethal or horrific, I think the risk-benefit ratio is clear.
yeah, I totally agree.
But don't you think we could do the same thing? Obviously, we can with AAV, just have a platform approval. There's been multiple studies now, right, with AAV2, AAV5, AAV8, AAV9, AAV1. It seems like we should be able to, leverage on the work that the safety profile of the AAV capsids. And then, of course, if there's a genetic disease, a monogenetic disease, we know the drug. It's the therapeutic gene that is going to correct the
Right.
defective gene. So it's not a lot of guesswork here. There's ways to streamline it. And, unfortunately, when this competitive world, you look at the Chinese, it is much quicker to get into clinical trials in
Yeah.
China than it is in the US.
Yeah,
And for these ultra rare diseases where, there's really no treatment options and they're devastating diseases, it seems that there would be a lot of benefit to decreasing the regulatory hurdles. So at
yeah.
least we give some hope to these kids and their families.
Is there some way
I
we can reduce the cost of manufacturing because that's another biggie, and it really costs a lot of money to build. So,
So we've done that. We've really increased, I think, our yield substantially with the cell suspension HEC 293 systems.
Mhm.
You know, I know at AskBio when we were pricing things, we could get the cost, less than 50,000, potentially in some cases to 10,000 per dose for some of our therapies. I would say that the costs aren't really that prohibitive. There's a big margin between the prices we charge and the costs of the drugs. I think a lot of times people think that the prices are being driven by the manufacturing costs, and I would say that's not the
Not
case.
the case, yeah.
Not the case. And you can
and
do things like doggy bone DNA, synthetic plasmid materials to further reduce the costs. I think there are definitely ways, and continuously increasing the yields would be beneficial. But eventually, right, we have to become like the monoclonal antibodies, and I think scientifically we know how to do that. It's just a matter of somebody putting in the investment and the time to make that occur. But I don't think the field is that far away from doing it. There are enough well-educated people in our sector who can make it happen. But with the dearth of investment right now, it's less likely to occur.
Yeah.
yeah, I mean, I would add too that I think if we could get capsids where we can give lower doses, that would help. But I also think too that there's a lot of variances between the manufacturers and how they actually manufacture and purify. And I think that as the science gets better and we whittle it down to like one or two types of manufacturing, that I think the technology will get better and the costs will be reduced even further. I mean, do you see that, Sheila? I mean,
Yeah.
I think that a lot of companies too, there's a lot of proprietary things that not everyone shares, and it's you can understand it from a corporate perspective, but I think eventually the manufacturing technologies will become more convergent, and it's like anything. Once more people are doing it in a specific way, the costs will come down.
Yeah.
Of course, the tariffs may
Yeah.
enhance the
Yeah.
cost.
Well,
Well,
I think
I
there is a lot of incentive for the industry to collaborate, right? I think there always has been, but especially now, there are a lot of competing technologies. There is a scarcity of resources. There's a lot of competition. You know, Jude and I made our manufacturing technology available for everyone through the Virogen enterprise, and so people could go there and essentially access our Pro 10 cell line. um, I know Jude has a lot of ideas on how to further increase the efficiency of manufacturing and drive down the costs, and I know others do too. And hopefully, there will be another renaissance in the field and we'll collaborate and overcome these problems because we have the capability, we have the scientific minds, and patients are still waiting, right? We've made some good proof of concept that this is an exciting technology platform. And I know everybody's getting a little tired. It's been a long haul, but it sure would be great if we finish strong and, overcome some of these barriers.
Yes. Well, guys, on that note, I think we're going to have to wrap up, but I think that this discussion has been really amazing and very, very exciting. So I look forward to major breakthroughs in the AAV field, which I'm sure will happen, and look forward to seeing you guys in New Orleans at the next meeting.